They always start with audience.
Hello everyone, Namaste. I'm Shrestha Bastula, researcher and public engagement manager at Research Institute for Bioscience and Biotechnology GRID here in Balkupari, Lalitpur. I would like to express my gratitude to all our participants and guests for joining us today for the 8th episode of Biomedicum seminar series. As a housekeeping rule, I would like to request you all to mute your microphone during the presentation, please. The presentation will run for 30 minutes and 15 minutes after the presentation is allocated for question and answer session. I would like to welcome and introduce our speaker for today's webinar, Dr. Raunak Shrestha. Currently, he's a postdoctoral research fellow at Helen Diller Family Comprehensive Cancer Center, University of California, San Francisco. Dr. Shrestha has done his PhD in bioinformatics from University of British Columbia, Canada. He has worked on numerous research topics leading to publications in high-impact journals. His research area include bioinformatics, cancer genomics, epigenomics, immuno-oncogenomics, machine learning, and many more. From starting his career at Kathmandu University to his PhD and further postdoctoral from Canada, he has achieved many prestigious awards and honors, one of which was the Post-Street Cancer Foundation PCF Young Investigator Award. We are very pleased to have him present today, and now I want to request Dr. Shrestha for his talk.
Thank you, Shrestha, for this kind introduction. And thank you, everyone, for joining the talk today. I'm just sharing my screen. Okay, can you see my screen?
Yes, we can.
Okay, yeah. First of all, thank you, RIBS and KRIBS team for inviting me to give a talk about my research. As Shrestha just introduced me, I am currently a postdoctoral research fellow at the UCSF in US, and also I am currently working as an adjunct research scientist in Nepal, and I will be talking briefly about like different research going on. So to start with, I will just talk about the work that I do. It's mostly in the cross intersection of cancer genomics, epigenomics, and infectious disease genomics. And I use different systems biology techniques and to work in different areas of precision medicine and understanding treatment resistance of different diseases, and try to build different bioinformatics algorithms and try to solve different challenging biological problems. And in this, I use different kinds of machine learning techniques, combinatorial optimization, linear programming. You'll get a taste of some of these in my talk today. And my main research interest lies especially in trying to contextualize different genomics data while making some clinical decisions. As you're all aware that there is an enormous amount of genomics data being generated now, and it is highly challenging to put all these genomics data into meaningful context and how we can utilize this a massive amount of data. So that's my main research interest, and whatever research I do, I put this particular text in mind and try to put all my research into a meaningful perspective. So I will give you a lot of different works that I've been doing, and all the works that I've been doing is to inform how can we integrate all this massive genomics data so that we can extract meaningful information that can be translated into clinic. So in today's talk, I will be briefly going over like two major studies that I've conducted. So the first study is mostly about a computational algorithm that prioritizes cancer driver genes. And this part of the talk will be more mathematical and more computationally heavy. And the second part of the talk, I will be giving about how we can utilize such complex computational algorithm to identify different cancer driver genes. And especially, I'll give you an example of a very rare cancer called malignant peritoneal mesothelioma, and how we identified cancer driver genes of this rare cancer that led to the identification of a therapeutic biomarker. And briefly, I will touch upon the different research that I have been leading in NAMI in Nepal, and that will be mostly in the context of infectious disease genomics, if time is permissible. So before that, I would like to start with this acknowledgement slide, and I would like to say that the talk that I'm giving today is mostly based on my PhD works that I did at University of British Columbia and Vancouver Process Center in Vancouver, Canada. And it is a work of a massive number of people and the collaboration at different institutes in North America and Europe. And during my PhD, I was mostly, my work was funded by MindTask and Prostate Cancer Foundation BC and the Canadian Institute of Health Research with the Bioinformatics training program at the University of British Columbia. So with that, I would like to start with my first part of my talk. That is, I will describe a computational algorithm called Hit and Drive that helps to prioritize cancer driver genes in individual patients. So in cancer, whenever, say, if we look at the evolution of a cell right from its inception, it accumulates numerous alterations. It could be mutations or different structural changes in DNA, and it accumulates those mutations. And at some point of the lifetime of the patient, some mutation might trigger cancer. And when we sequence these cancerous genome and we try to identify different alterations going on in these cancer patients, we identify numerous alterations, like hundreds to thousands of mutations. But most of these mutations does not provide any growth advantage to the tumor. And those mutations or alterations are termed as passenger alterations. And there are very few alterations that actually drive the cancer. There may be like some mutations that initiate the cancer, and when there is a treatment given, some of the mutations might be responsible for the treatment resistance, and it might help in the progression of the cancer to the advanced form and so on. So there are very few driver alterations that are positively selected during the tumor evolution. And those driver alterations are the causative agents of cancer. And that those driver alterations are important because we can target those driver alterations in the clinic, and so that we could treat and manage the patients. So that's why important identification of these driver alterations are very, very critical for the treatment of cancer. But the main challenge is that these driver alterations are diluted and outnumbered by the vast amount of passenger alterations, making the identification of these driver alterations very complicated. So whenever we sequence these cancer genomes, we get lots of different mutations. And if we just plot the distributions of these mutations in terms of how many times we observe those mutations across different patients, we get this kind of distributions as shown in the slide, and where the vertical axis represents the number of patients that harbor an individual mutations. And in the horizontal axis, there are different genes laid out which are mutated in cancer. So we can see that there are very few genes that are recurrent or that are present or mutated across a large fraction of patients. For example, TP53 is prime example where it is mostly commonly mutated in a number of different cancer patients. But if we look at the other genes, it is hardly mutated in very few fraction of patients. So it is very easy to identify highly recurrent mutations. And of course, those are important mutations, but there are also a long tail of different mutations that might be functionally important and that might be present in a very small fraction of patients, but could have a meaningful impact in driving those cancers. So it is very critical to identify these long tail of mutations, and identifying these mutations are very, very difficult. So to address these challenges of identifying driver mutations, during my PhD, I worked on a couple of papers describing an algorithm called hidden drive, that helps to prioritize this rare as well as most recurrent driver genes. And the initial version of this paper was published back in 2014 in RECOM conference, and we improved that our algorithm to a larger extent and published with additional analysis in 2017 in Genome Research. And I will be talking about this work today in my talk. So talking about the main problem that we are trying to address is that whenever we sequence cancer genomes, we observe different mutations and different genes. And we are interested to know which of those genes are cancer driver genes that are responsible for driving cancer. Now, we know that these genes does not act in isolation. Like one gene interacts with different other genes, forming an interaction network. And this collective information can be represented in terms of a network. So in the picture here, I have shown this schematic visualization of a network where individual circles are the individual genes. And the link between those genes, or you can say these circles are genes or its translated product, that is proteins, and the link connects one gene to the other gene, or you can say one protein to the other protein, and the link links represents the physical interaction between those proteins. So this is a protein-protein interaction network where we are trying to visualize the interaction between one protein to the other protein. And the red circles represents all the mutations or structural aberrations that we observe in cancer patients. So now we are interested to identify the location of these driver genes within the context of this interaction network. So that is the main problem we are trying to address using our algorithm. So we came across this a very classic algorithm called facility location problem. So this facility location problem was invented somewhere around 1800s, and it is concerned with optimal placement of facilities to minimize associated cost. Now you can think of these facilities as buildings or campuses or some hospitals or say or restaurants. And there are also clients that surround this facility. Now, this problem, facility location problem, is concerned with placement of this facility in such location such that all the clients can have equal access or easy access to these facilities. Now, this problem has been applied to numerous different real world problems, for example, hospital placement or airline hub, airport selection, or say Starbucks supply chain operation, FedEx supply chain operation, and even it can be applied to different social media research. For example, if you want to place a fake news or bot, like a bot, you can use this algorithm to identify which location we can would be most optimal to place those bots. So this problem has been applied to many real world contexts. Now, all the associated variables within these, problems are more predictable, but in biological contexts, we have a different set of challenges. Now, when we look at the biological network, we have a source that is it could be a mutated gene, which could be a causative agent of cancer. So we'll call it as a source, which has some mutations or structural aberrations that generates some kind of signal. And this signal is transmitted to some distant genes. And this distant gene, we call it like a target, or it could be an outlier gene, which changes its expression. So the expression or the activity of this distant gene is changed in some way by because of this source gene. Now we can model this problem as the shortest path problem, because if we want to measure the influence of the source to the target, we can just assume that it will take the shortest path in the network. But our biological molecules are non-intelligent, so it cannot compute the shortest path. So there is a lot of uncertainty in the biomolecular interactions. So we try to model this as a random walk process and measure the influence as the hitting time distance. So in the hitting time distance is basically the number of steps that it takes to travel from the source to the target. So if we look at the hitting time of this figure A, it is just 1, 2, 3, 4 hops. So in 4 hops, we can reach from the source to the target. So that's the hitting time. And the inverse of this hitting time will give us some kind of measure of the influence. So So in context of cancer, they're not just a single driver gene. There could be multiple genes that are mutated at the same time, and all those mutated genes generates a signal, some kind of biological signal simultaneously, which might affect a particular target. So in that context, we could model this as a random walk process, and we measured using the something like hitting time using a multi-hitting time, where this the multiple source influences the distant target, but we measure whichever random walk reaches the target genes first. So that's the multi-hitting time. So we formulated our problem as the random walk facility location problem, where we want to identify the smallest set of these driver genes, which can maximize the multi-hitting time. That is, which can have the maximum impact in the changes of the expression of different clients or the different other distant genes. But this, to compute the multi-hitting time, it is very, very hard problem. It is an NP-hard problem. That means it cannot be computed in a polynomial time. So we cannot use the computational tools or power at our disposal to compute this multi-hitting time. So in order to account for that, we reduce this problem into a weighted multi-set cover problem, where we try to identify the smallest set of driver genes that can have a large influence in the changes in the expression of those outlier genes. So that's how we reduce the complexity of the problem. And this is the overview of the algorithm that we built. So we have different sequence alterations data that comes from the DNA sequencing. We could have mutations or changes in copy number or gene fusions within individual cancer patients, and we can represent these information as a binary matrix of patient versus the genes. So we can tag which genes are mutated in individual cancers. And for the same patient, we profile the expression or the activity of individual genes, and we can identify which of those genes are changed or which of the genes are differentially expressed. So these information can also be represented in the form of a binary matrix of patients versus the genes. And then we can bring in the information about the interaction matrix of how a particular one gene can influence a distant gene in the network, and we can calculate the pairwise influence of one gene to the other, and then we can use all these three genes all this information from these three sources into our algorithm and try to compute for individual cancers which of those genes are responsible, or which of those the smallest set of genes that can have a maximum impact on changing the activity of the distant genes. So that is how we try to prioritize the cancer driver genes. And in doing so, we model this this problem as a integer linear programming. So I will just give you a crash course of the what linear programming is. I believe like every one of us has studied this linear programming during our high school, where we have we are given multiple set of equations of a straight line, and we are interested to identify this solution space for solving different values of x and y's and so on. So this is a linear programming where we can identify what is the value of x and y and the solution space. And the integer linear programming is just that the values of the x and y can be an integer and not any other number outside integer. So we used integer linear programming, and this is just a different set of equations that we modeled with. So all these variables like x and y's, these represent different stuff within our algorithm. I will not go into detail, but individual genes and all the mutated genes are represented by x, and all the outlier or the changes in the genes that have changes in gene expression are represented as y. So for every gene, we will have a separate equation. So when we generate all these possible equations, we have equations for millions to billions, depending on the how many patients we have and how many mutated genes and changes in gene expression we have. So we will be generating millions to billions of equations, and we are solving those millions of equations of those straight lines to come up with a solution to identify the smallest set of mutated genes that can have a maximum impact in changing the expression of those within those patients. And those patients, those genes are identified as the cancer driver genes. So after building this algorithm, we applied this algorithm to multiple different cancer data sets that are publicly available. So we used, for our analysis, we used 4 different cancer types: glioblastoma, that is the brain cancer, as ovarian, prostate, and breast cancer. And what you are looking at is a results of more than 2,000 patients across these 4 cancer types. And for all these 2,000 cancer patients, we identified cancer driver genes based on the integration of the genomic or the DNA level data with the RNA level data. And we try to identify all the cancer driver genes. And we try to measure if we can predict some targeted therapies or some drugs that are able to inhibit the function of those cancer driver genes, and if our algorithm is working or not. So that was what we were trying to address. And we looked at different drugs that are either clinically approved, that are drugs that are in clinical trial or preclinical stages, and we identified that in most of the cancers, we were able to identify cancer driver genes that had some drugs that was able to predict, was able to inhibit those cancers. So those driver genes we identified using our algorithm, we could use it to identify which genes should be targeted for an individual cancer patients. And further, we also wanted to test the efficacy of individual cancer drugs. And we found a data set from a published paper, from this paper published in Cell 2016. And they had profiled about 1,001 cancer cell lines from 30 different cancer types. And all these cancer cells were treated with 265 different drugs. These drugs were FDA approved, there was targeted or cytotoxic drug, there was FDA approved or drugs in a clinical trial or preclinical stages. And all these cell lines, more than 1,000 cell lines, were treated with 265 drugs. So we have more than 265 combinations, or you can think of as we are analyzing data for about 265,000 cancer patients. And each of those cancer cell lines had masked genomic and the transcriptomic data. And we used this data to identify cancer driver genes. And these individual cancer cell lines were also screened for their sensitivity. So individual cell lines were either categorized as sensitive to the drug or resistant to the drug. And we were interested to identify or predict the drug efficacy and different, if these driver genes are informative in predicting the drug efficacy. So for that, we tried to use one of our previously built algorithm from our lab called OpDis, which where we start from the driver genes and we seed the driver genes in the network, and we try to identify sub-networks of genes, that is small connected networks of genes, which can collectively discriminate the sensitive cell lines versus the resistant cell lines. And these subnetworks can efficiently classify the responsive versus the non-responsive cell lines. And I will just summarize the results of what we found. So we looked at different drugs and within the different cancer contexts. So this results just summarizes the three of the results for three of the drugs. So one is the geftinib, that is used as an EGFR inhibitor for breast cancer. Other is the telazolomide in the context of glioblastoma, and the Neutralin 3A in the context of ovarian cancer. So we know that geftinib is a inhibitor for EGFR, and we predicted that EGFR, that is the driver gene that we predicted from those analyzing cancer cell lines, accurately predicted the responsive versus the non-responsive. And the same goes for the other two drugs, where we identified the intended target of the drug. In addition, we also identified different other genes and the proteins that are involved in the targeted pathway. For example, we identified different genes involved in this EGFR pathway. And for this telazolomide, we also identified different targets of this pathway over here. So using our approach, we could identify not only the intended target and not only the efficacy of the drugs, but also different other genes that are involved or that are dysregulated when this a particular drug is given to a patient or given in the context of cell lines. So we could identify those different genes involved in the drug treatment for different kinds of analysis. So there are numerous different analyses that I've done that are present in the paper, and I've just highlighted a couple of these analyses. So in summary, so we have developed an algorithm, Hidden Drive, which collectively measures the effect of multiple different driver genes and which has the maximum impact on the changes in the expression of the target activity of the target genes. And in this way, we can help contextualize this multi-omics data while making treatment decisions. So this is the summary of what we have done in this development of this novel cancer driver prediction algorithm. So having developed this algorithm, I was interested to apply this algorithm to a novel cohort of cancer patients. So now I will jump into another part of our talk where I used a rare cancer, cohort of cancer called malignant peritoneal mesothelioma. And we applied this algorithm, Hidden Drive, to identify driver genes and the therapeutic biomarker. And this study was published back in 2019 in Genome Medicine. So to introduce about the malignant peritoneal mesothelioma, so this is a very rare cancer that arises in the lower abdominal cavity, mostly in women, but also men also get it, but it's mostly in women. So this mesothelioma arises around the peritoneal lining of the like lower abdomen. All this lower abdomen is covered by a very thin transparent membrane known as the mesothelial tissue or the mesothelial lining, and this cancer arises around this area. And this is a very rare form of cancer. So most mesothelioma cancer arise in lung, which is called as the pleural mesothelioma. And I am describing about the peritoneal mesothelioma, which accounts for about 20 to 30% of this for all cases of mesothelioma. But currently, the standard of care is just the cytoreductive surgery followed by chemotherapy. So if a patient is presented with the peritoneal mesothelioma, the doctor performs surgery, and immediately after that, they perform chemotherapy. So that's the standard of care till date. But these chemotherapy are not effective, and they are not known targeted therapies still at the moment for majority of the mesothelioma. So we were interested to identify if we can predict any possible driver genes within this rare cancer. So we started with this project and we tried to reach out to different study, different others like groups around North America and Europe. And since this is a very, very rare cancer, we had a very hard time collecting the tissues, and we were able to collect about 19 different tissues from 19 different patients. And this is the largest cohort of peritoneal mesothelioma tissues that were sequenced for both genome, proteome, genome, transcriptome, and proteome. And this is the largest cohort in the world that exists till date. And this is, believe me, this is just 19 cases, and it's a very rare cancer, and it is the largest cases of patients that available for this patient for this cancer type. So we got access to this 19 patients' tissues, and we performed DNA sequencing, RNA sequencing, and we performed protein analysis, proteome analysis using mass spectrometer. And we integrated these all data coming out from individual 19 patients using the algorithm that I just described, and tried to identify potential driver genes that are present within these cancer types. So after we use this algorithm, we identified a gene or a collection of 4 genes called BAC1, PBRM1, ZD2, and SMARCC1, all a part of chromosome 3p21, had like a deletion in almost half of the cancers. So this is a plot summarizing the copy number profile of those patients, all those 19 patients, in terms of the percentage. The red indicates the gain of copy number, and the blue indicates the loss of copy number across the entire genome. The plot on the bottom is the same plot, but segregated by individual cancer patients. And we identified this gene this locus in chromosome 3p21 that was deleted in almost half of the cancer. And in this study, I will present several evidences that indicate that these patients which have deletions of BAP1 gene, or this chromosome 3p21, forms a distinct subtype of peritoneal mesothelioma. And even a single copy loss of this BAP1 gene is sufficient to classify the those genes into a separate subtype. So there are so I will present evidences that there are 2 different subtypes of this cancer, one with the BAP1 deleted, which we call as BAP1 deletion, and the other with the BAP1 intact, that is the as highlighted in the yellow here. So first, we measured the gene expression of these cancer, and also in context of their copy number. So I have laid out these 19 patients here, and we are visualizing the mRNA expression using the RNA sequencing data, and the color represents the red represents copy number gain, which has the highest expression over here, and the copy number neutral, that is if that is diploid. This is the mRNA expression level, and if there is a copy number loss, whenever there is a copy number loss, there is decrease in expression of this BAP1 gene. And even if there is a homozygous loss, that is the both copy of the gene was lost, the expression level was even suppressed. And this was at the mRNA level. We also validated this at the level of protein, where we conducted IHC staining on the tissue for the BAP1 nuclear staining. And you can see for the BAP1 copy number gain, there was lots of staining for BAP1, and this is for copy number neutral, and this is for heterozygous and homozygous loss, where there was no or very less staining for the BAP1. So this indicates that there is a allelic loss of BAP1 is correlated with decreased transcript and protein level expression of this corresponding gene. And we also looked at the copy number data in context of different signaling pathways. And here you're looking at the individual cancers and the individual genes categorized into different pathways, where the red color indicates the copy number gain and the blue represents copy number loss. So you can see, especially it affects the Swiss-SNF complex genes that are deleted in almost half of the cancer cohort. And there is also genes related to chromatin remodeling and DNA repair pathway, and some cell cycle pathways are altered in this BAP1 deletion subtype. We also looked at the mRNA and the protein expression of individual cancers in the context of different signaling pathways, and there were differences in different pathways, for example, chromatin remodeling, DNA repair, cell cycle pathways. But strikingly, we found that there was changes in immune system-related pathways. For example, the cytokine signaling and the innate immunity was activated in the backbone deletion patients, and whereas for the backbone impact cases, the MHC presentation and the adaptive immunity pathways were activated, and these immune system pathways were more active in the backbone deleted cases. So we thought of if there is a immune infiltration within these backbone deleted cases. So in order to test whether there was immune cell infiltration within these backbone deleted tumors or not, we tried to measure the immune score and the stromal score. We correlated this using the gene markers published from this study here. And you're looking at the correlation of this stromal and immune score using the mRNA expression and the protein expression here. Individual dots represents individual cancers that are and the color represents the different subtype, backbone deleted and the backbone intact, and the correlations of this immune score and stromal score are very strong in these both mRNA and protein level, which potentially indicates that there is potential leukocyte infiltrations or the immune cell infiltration from the tumor microenvironment. So this led us to another question: if we can identify those different immune cells infiltrated within these cancer types. So for this, we measured the presence of different immune cells using the gene expression data, and we used CyberShot software where it leverages the gene expression signatures of these individual immune cells and try to estimate the proportion of different immune cell types that are present in the individual cancer types. So especially we can see that there is a massive infiltration of T cells that is highlighted in red over here, and the B cells that are highlighted in the gray color. So T cells and B cells were massively infiltrated in a majority of the tumors. And even we validated this using the IHC staining of the CD3 and CD8 markers that are the markers for the T cell infiltration. So we could see staining for this T cell infiltration within this tumor. So this strongly indicates that there is a immune cell infiltration in this peritoneal mesothelioma cases. And finally, yeah.
Sorry, since we are running out of time, I would like to request you to.
Okay, this is my last slide over here.
Thank you.
Okay, so we finally identified that there is changes in different gene expression of different immune markers, especially LAG3, ICOS, CTLA4, PD1, PD-L1, and where there are inhibitors present for these markers, and those category of drugs are known as the immune checkpoint blockade therapies or drugs. And so this analysis indicates that there is a high expression of these markers for backbone deleted cases, so we can drug these patients with the immune checkpoint blockade therapies or immunotherapies. So in summary, we identified a subtype of this peritoneal mesothelioma cases where we identified like different immune checkpoint markers, and potentially these patients can be recommended for immune checkpoint blockade therapies. So I think if we have like a couple of minutes, I can just go over some of the research. I have just a couple of slides if the time allows. Is that okay?
Okay, you can go forward.
Okay, so I will just highlight what I've been doing in NAMI in Nepal and a couple of projects. So the first project I'm working at, looking at is in the infectious diseases and particularly in the tuberculosis. And what I'm doing here is trying to identify different drug-resistant mutations within this tuberculosis. And we have this is a collaboration between me and Dr. Bisis Khanal, and we are trying to mine a large cohort of publicly available whole genomes of this Mycobacterium tuberculosis to identify different drug-resistant mutations using deep learning techniques. And this is some ongoing work that is led by Parikshit Prasai, and this study has been funded by National Institute for Health Research in the UK and RSHTM. And this is one of the work. I will not be talking about any details of the work. And another work we are doing is trying to look at the evolution of COVID-19, SARS-CoV-2 in Nepal and in different other countries, and try to leverage different genomics data that are present. And this is also an ongoing collaboration between me and Dr. Bisis Khanal, and the study is led by Saurabh Baral. And we are trying to look at how this COVID-19 virus has evolved in Nepal and how it's transmitted, and how can we develop different tools and techniques in order to prevent future pandemics. So these are some of the ongoing works that I've been working at. Probably in the future talk, I will be able to present some interesting results coming out of these studies. So with that, I would like to conclude my talk for today. So I'd be happy to answer any questions. And I'd like to apologize that I went over time. Thank you. Yeah, if there's any questions, yeah, I would like to answer.
Yeah, now the floor is open for questions. You can either ask by unmuting or send your question through chats, please.
Shall I start? So Raunak, yeah. सबै नेपाली नै रहेछ उसमा हैन बक्समा नेपाली क्वेशन्सहरु हुन्छ
ओके
ओके मेरो 4 वटा क्वेशन्सहरु थियो हैन जस्तै अघि तिमीले भन्यो ड्राइभर जिन्सको बारेमा हैन एउटा भनेको EGFR को कुरा गर्यो हैन एउटा चाहिँ अब जस्तै नेपालमा पनि यहाँनिर क्यान्सर लाग्यो कसैलाई भनेर डायग्नोसिस गर्न गयो भने चाहिँ आजकल PCR को टेस्टिङ गर्छ के हैन अनि तिनीहरुले पनि EGFR को म्युटेसन सुरुमा हेर्दै रहेछ के तिनीहरुले यहाँ पनि हैन त्यसै गर्दाखेरि जुन यहाँ नेपालमा लागू भइरहेको छ अथवा यो त पहिलेदेखि बाहिरबाट आएको टेक्नोलोजी होला अनि त्यो बेसिकली ड्राइभर जिन भएर नै गरिरहेको हो कि अब अरु पनि यसको कुनै कज छ EGFR को हेर्नुको कारण
अब EGFR को कुरा गरे हैन EGFR चाहिँ अब एउटा cancer driver genes, especially यो चाहिँ lung cancer, other like other different cancers are present. Mostly lungs cancer or EGFR, it's a prime driver cancer mutations हो र यो driver gene मात्रै होइन कि त्यो driver gene लाई चाहिँ inhibition गर्ने drugs already available छ हैन so त्यो चाहिँ एकदमै low hanging fruit भएकोले चाहिँ यो EGFR mutationsहरु चाहिँ सुरुमै चेक गरिन्छ किनभने यो चाहिँ लङ जस्तै लङ क्यान्सरको कन्टेक्स्ट या अरु प्यान्क्रियाटिक क्यान्सरको केसेसहरुमा चाहिँ यो EGFR अरु यस्तै केसेस इभन ब्रेस्ट क्यान्सरमा पनि कहिलेकाहीँ हुन्छ मेजोरिटी अफ द पपुलेसनमा चाहिँ यो EGFR म्युटेसन पाइन्छ सो त्यो भएको कारणले चाहिँ डाक्टर्सहरुले चाहिँ EGFR म्युटेसन चाहिँ सुरुमा चेक गरिन्छ हैन तर यसको चाहिँ एउटा ड्रब्याक के हुन्छ भन्दाखेरि यदि अब भनौं कुनै पेशेन्टलाई चाहिँ EGFR म्युटेसन भएको छैन भनेर पत्ता लगाए हैन त्यो भएको केसेसमा चाहिँ EGFR को इन्हिबिटर चाहिँ दिन मिलेन त्यो पेशेन्टलाई हैन तर अरु जिन्सहरुले चाहिँ त्यो जिन्सलाई ड्राइभ गरिरहेको हुन सक्छ हैन अनि त्यसैले चाहिँ हामीले लाइक अब होल एक्सोम या होल जिनोम सिक्वेन्सिङहरु गरेर कुन चाहिँ जिन हो कुन चाहिँ अरु ड्राइभर हो भनेर पत्ता लगाउनु पर्ने हुन्छ
ओके अनि यो ड्राइभर पत्ता लगाएपछि चाहिँ यो चाहिँ डायग्नोस्टिक टुलको रूपमा युज हुन सक्छ
डायग्नोसिस भन्दा पनि यो थेराप्युटिक भनौँ न अब अफकोर्स अब डायग्नोस्टिक्स पनि हुन सक्छ हैन तर मोस्टली चाहिँ यो डायग्नोसिस भइसकेपछि चाहिँ अब होल जिनोम सिक्वेन्सिङहरु गरिन्छ हैन अनि कुन म्युटेसन छ भनेर चाहिँ एनालाइज गरिन्छ त्यसपछि अब कुनै जिन्सहरु त्यस्तो पत्ता लगायो या कुनै जसको विरुद्धमा चाहिँ इन्हिबिटर्स ड्रग्सहरु अवेलेबल छ भने त्यो जिन्सहरु या त्यो ड्रग्सहरुलाई चाहिँ रेकमेन्डेसन गरिन्छ त्यो पोटेन्शियल ट्रिटमेन्ट क्यान्डिडेट्स हुन सक्छ भनेर
ओके अनि जस्तै थ्यांक्स अनि अर्को जस्तै यो कोलन क्यान्सरमा पनि यो केरास म्युटेसन हेर्छ नि हैन यो पनि त्यस्तो कुरा भयो
हो एक्ज्याक्टली त्यही यो ड्रग्सको अगेन्स्टमा चाहिँ सरी त्यो जिन्सको अगेन्स्टमा त ड्रग्स भएकोले नै यिनीहरूलाई चाहिँ सुरुमै चेक गरिन्छ हैन र पपुलेसनमा धेरै पाइने भएकोले पनि यो चाहिँ फर्स्ट लाइन चाहिँको त्यो ट्रिटमेन्टको लागि चाहिँ युज गरिन्छ
ओके अनि रौनक स्लाइड नम्बर 19 हेरे मिल्छ फेरि Slide number 19.
19. Okay. Share about this one. Okay. Yeah.
Okay. यहाँनिर जस्तै यत्रो म्यासिभ यो ड्रग स्क्रिनिङ भएको छ नि यहाँनिर अनि यो त सबै रोबोटले गर्ने होला
यो सेल लाइनको स्क्रिनिङहरु चाहिँ रोबोटले गरिन्छ या
ओके अनि यो सबै सेल लाइन्सहरु चाहिँ आफै डेभलप गर्छन् कि यो कमर्सियल सेल लाइन्सहरु सबै यिनीहरु चाहिँ
यो सेल लाइन्सहरु चाहिँ अब यो चाहिँ अब स्याङ्गर इन्स्टिट्युट र ब्रड इन्स्टिट्युट मिलेर चाहिँ यो चाहिँ एउटा प्रोजेक्ट गरेको छ हैन यो त म्यासिभ लाइक इन्टरनेशनल कोलाबोरेटिभ प्रोजेक्ट हो हैन अनि उनीहरुले चाहिँ यो सेल 2016 मा चाहिँ उनीहरुले पब्लिश गरेको डेटा छ र उनीहरुले सबै डेटाहरु चाहिँ पब्लिकली फ्रीली अवेलेबल भनेर चाहिँ शेयर गरेको छन् हैन अफ कोर्स यो सानोतिनो इन्स्टिट्युसनले चाहिँ गर्न सक्दैन हैन उनीहरुको लाइक कोलाबोरेसनले सबै यो 1001 डिफरेंट क्यान्सर सेल लाइन्सहरु कलेक्सन गरेर अनि यो लार्ज लिस्ट अफ ड्रग्स बाट ट्रिटमेन्ट गरेर त्यो डेटाहरु चाहिँ पब्लिक डोमेनमा राखेर उनीहरुले चाहिँ फ्रीली अवेलेबल गरेको छ र त्यो डेटालाई चाहिँ डाउनलोड गरेर हामीले प्रोसेस गरेर एनालाइज गरेर हामीले चाहिँ हाम्रो स्टडीमा चाहिँ युज गरेको तर हामीले एक्सपेरिमेन्ट चाहिँ हामीले गरेको हैन यो चाहिँ स्याङ्गर इन्स्टिट्युट र ब्रड इन्स्टिट्युटको कोलाबोरेसन उनीहरुले चाहिँ गरेको
तिमीले चाहिँ पब्लिक डेटा युज गरेको
या
ओके अनि जस्तै रौनक लाग्छ जस्तै अघि रौनकले जुन त्यो नामीसँगको कामको कुरा गरेको जुन मल्टी ड्रग रेसिस्टेन्ट ट्युबरक्युलोसिसको म्युटेसनहरु पनि हेर्यो नि त्यो पनि पब्लिक डेटाबाटै तिमीले एक्सट्र्याक्ट गरेर के छ भनेर हेर्न खोजिरहेको खासमा हैन
या हामीले चाहिँ अब अफ कोर्स नेपालमा अहिलेसम्म अब ट्युबरक्युलोसिसको जिनोम धेरै सिक्वेन्स भएको छैन आई थिंक भेरी रिसेन्टली एउटा या दुईटा नेपालबाटै कलेक्ट गरेको क्लिनिकल आइसोलेट्सहरु चाहिँ सिक्वेन्सिङ भएको छ हैन तर त्यो भन्दा बढी भएको छैन तर पब्लिक डोमेनमा हेर्ने हो भने ट्युबरक्युलोसिसको चाहिँ म्यासिभ डेटा अलरेडी अवेलेबल छ हैन अनि कति चाहिँ इभन साउथ एसियाबाट पनि धेरै छ इन्डिया चाइनाबाट हैन अनि लाइक युरोप नर्थ अमेरिकाबाट धेरै छ र हाम्रो चाहिँ अब इन्ट्रेस्ट भनेको चाहिँ अब नेपालमा बायोइन्फर्मेटिक्स रिसर्च कसरी स्टार्ट गर्ने हैन अनि यसलाई हामी कसरी अगाडि बढाउन सक्छौं अब अहिलेको कन्टेक्स्टमा चाहिँ नेपालबाटै अब डीएनए सिक्वेन्सिङ गर्नको लागि अब धेरै त्यो फन्डिङहरु रिसोर्सेसहरुको च्यालेन्जेस छ हैन त्यो एक ठाउँमा भयो तर हामीले अहिले के यो पब्लिक डेटाहरुलाई युज गरेर नेपाललाई नै युजफुल हुने रिसर्चहरु गर्न सक्छ सक्दैन हैन त्यो चाहिँ हाम्रो अहिलेको मेन अब्जेक्टिभ हैन त्यसैले चाहिँ हामीले लाइक पब्लिक डोमेनमा भएको ट्युबरक्युलोसिसको डेटाहरु डाउनलोड गरेर अलमोस्ट हामीले मोर देन आई थिंक 8000 10000 आइसोलेट्सहरुको चाहिँ डेटा प्रोसेसिङ गरिसकेका छौं हैन अनि यो चाहिँ सबै होल जिनोमबाट डाउनलोड गरेर दे आर फ्रीली अवेलेबल एन्ड हामीले प्रोसेस गरेर चाहिँ हामी वी आर वर्किङ अन इट
ओके अघि तिम्रो स्लाइड नम्बर 19 र यो नानीको कुरा गर्दाखेरि नानी गरेको कामको कुरा गर्दाखेरि यो सबै पब्लिक डेटाबेसबाट तिमीले त्यहाँबाट भएको कुरा एक्स्ट्रा गर्न खोजेर चाहिँ गरिरहेको काम देखियो तर यस्तै काम वेट ल्याबमा जस्तै मानौ त्यति तिमीले भने जस्तो त्यति धेरै सेल लाइन्स नहोला त्यति ड्रग क्यान्डिडेट नहोला नेपालमा त्यस्तो काम गर्न सम्भव छ होला अक्सर हुनको लागि
like of course like
ड्रग स्क्रिनिङमा चाहिँ
like प्रजलले पनि अब क्यान्सर सेल लाइन्सहरुमा काम गरिरहेछ हैन you know the challenges more than me हैन अनि of course अब नेपालमा चाहिँ अब यो ड्रग स्क्रिनिङहरु गर्न चाहिँ like some level सम्म सकिएला कुनै एउटा सानो स्केल सम्म चाहिँ if we have the appropriate resources हैन like अब कुनै त्यो ड्रग स्क्रिनिङहरुमा युज गर्ने कुनै अटोमेटेड मेसिनहरु हुन्छ हैन यदि अब त्यस्तो मेसिनहरुसँग एक्सेस या त्यस्तो मेसिनहरु चाहिँ हामीले नेपालमा भित्र्याउन सक्यौं भने चाहिँ मेबी हामी एउटा सर्टेन लेभलसम्म चाहिँ हामी गर्न सकौँला तर मेरो चाहिँ मेन क्वेशन चाहिँ के हुन सक्छ भने त्यो हैन हाम्रो मेन अब्जेक्टिभ के हुन्छ यदि त्यो कन्टेक्स्टमा हैन हामीले नेपालमा ड्रग स्क्रिनिङ गरेर त्यसलाई नेक्स्ट भनौं ड्रग स्क्रिनिङ गर्यौं भने त्यसको नेक्स्ट स्टेप के हो हैन that is the most challenging part हैन हामीले कसरी त्यो ड्रग स्क्रिनिङबाट आएको कुराहरु हामीले युटिलाइज गर्न सक्छौं हैन त्यो एउटा च्यालेन्जिङ एस्पेक्ट भयो तर म चाहिँ लाइक अब आई वान्ट टु ओपन द फ्लोर टु एभ्रीवन हैन यो क्वेशन चाहिँ आई थिंक अहिलेको कन्टेक्स्टमा चाहिँ इन माई ओपिनियन हैन हामीले चाहिँ नेपालको कन्टेक्स्टमा चाहिँ what we need to understand is हामीले आन्सर भन्दा पनि क्वेशनलाई चाहिँ हामीले बढी फोकस गर्नुपर्छ के हामीले कुन क्वेशनलाई चाहिँ हामी एड्रेस गर्ने हो जुन चाहिँ नेपालको कन्टेक्स्टमा एकदमै फिजिबल हुन सक्छ हैन एन्ड व्हाट क्वेशन इज इम्पोर्टेन्ट टु आन्सर इन नेपाल हैन जस्तै अब नेपालमा हामीले हाम्रो अब डिफरेन्ट च्यालेन्जेसहरु छ रिसोर्सेसहरु एकदमै कम छ हैन अनि एक्सपर्टिजहरु देखि लिएर फन्डिङहरुको एकदमै अभाव छ सो इन दैट कन्टेक्स्ट के हामीले नेपालमा क्यान्सर रिसर्च गर्ने हो भने बाहिर लाइक अब विदेशमा गरेको रिसर्चहरुलाई नै त्यसको रिजल्ट्सहरुलाई लिएर हामीले एप्लाई गर्ने हो कि नेपालकै केसेसहरुमा भनौं अब सिक्वेन्सिङ गरेर या कुनै वेट ल्याब एक्सपेरिमेन्ट गरेर नेपालको कन्टेक्स्टमा त्यसरी डेटा जेनेरेसन गर्ने हो हैन दैट वी नीड टु अन्डरस्ट्यान्ड फर्स्ट हामीले कुन क्वेशन चाहिँ एड्रेस गर्न सक्छौं या कुन क्वेशन्सहरु चाहिँ एड्रेस गर्न इम्पोर्टेन्ट हुन्छ हैन त्यो चीज पत्ता लगाउनु पर्यो हैन फर एक्जाम्पल अब जस्तै कि एउटा अब पपुलेसन लेभलमा हामी कुरा गर्ने हो भने नेपालीहरुलाई लाग्ने क्यान्सर विदेशीहरुलाई लाग्ने क्यान्सर भन्दा फरक हुन्छ कि हुँदैन हैन र अब फर एक्जाम्पल जस्तै कि अब प्रोस्टेट क्यान्सरको कन्टेक्स्टमा हेर्ने हो भने अहिले लाइक अब चाइनिज पेशेन्ट्सहरुमा लागेको प्रोस्टेट क्यान्सर र युरोपियन र नर्थ अमेरिकन लाई लागेको क्यान्सरहरुमा देयर आर सर्टेन मोलिकुलर डिफरेन्सेस छ भनेर पत्ता लगाएको छ हैन तर अब त्यस्तै अब स्टडीज गर्ने हो भने नेपालमा भनौं न इट कुड बी लट अफ च्यालेन्जिङ सो हामीले चाहिँ अहिले के कुरामा चाहिँ हेर्नुपर्ने आवश्यकता छ भने अहिलेसम्म एभाइलेबल डेटाहरु फर एक्जाम्पल क्लिनिकल डेटाहरुमा चाहिँ जस्तै कि कुनै भनौं न सपोज कुनै क्यान्सर पेशेन्टलाई ट्रिटमेन्ट गरेको छ भने अब विदेशीहरुलाई अब ट्रिटमेन्ट गर्दाखेरि यस्तो रिजल्ट देखियो नेपाली पेशेन्टहरुलाई ट्रिटमेन्ट गर्दाखेरि अलि फरक देखियो रिजल्ट्सहरु हैन क्लिनिकल भेरिएबललाई मात्र हेर्ने हो भने हैन त्यस्तो कन्टेक्स्टमा चाहिँ वी क्यान प्रायोरिटाइज दोज काइन्ड अफ रिसर्च अब यदि अब हामीले नेपाली पेशेन्टहरुलाई पनि ट्रिटमेन्ट गरेको रेस्पोन्स र विदेशीहरुलाई गरेको क्यान्सर पेशेन्टहरुलाई गरेको रेस्पोन्स उस्तै छ भने देयर माइट बी सिमिलरिटी इन द बायोलोजी हैन सो त्यस्तो केसेसहरुमा चाहिँ हामी बाहिरै भएको रिजल्ट्सहरु र बाहिरै भएको त्यस्तो ड्रग स्क्रिनिङ त्यस्तो रिजल्ट्सहरुलाई नेपालमा एप्लाई गर्न सकौँला तर यदि नेपालकै कन्टेक्स्टमा एउटा भिन्न रिजल्ट आएको छ भने आई थिंक इन दैट कन्टेक्स्ट नेपालमै यस्तो ड्रग स्क्रिनिङ गरेर नेपाललाई चाहिने के हो भनेर चाहिँ हामी त्यो क्वेशन्सहरुलाई एड्रेस गर्न चाहिँ हामी लाइक अब ग्रान्ट लेख्नुलाई पनि अलिकति मेबी वान अफ द मोटिभेसन फ्याक्टर हुन सक्छ
थ्यांक्स रौनक
Yeah, any other questions?
I think that's all the questions for today. Yeah, it's been a pleasure being with you all today, and I would like to appreciate the effort and time of our guest speaker, Dr. Raunak Shrestha. We hope that today's talk have been informative to our participants. So now, thanking you all, I would like to conclude today's webinar.
Thank you, K-Ribs, Prajol, and Subiksha. Thanks for inviting. It was pleasant talking with you all. Thanks a lot.
Thank you.
Okay, bye-bye.
Thank you. Bye.